Hello, everyone. A very good evening to all of you who are joining us here today. So, welcome to the PhD Talks by NcodeX Episode 8. For the program today, we have a very talented speaker. By name, he is Mr. Qambar Hasan, and he works as a technical officer and a teaching academic at Center for Cellular and Molecular Biology, Life and Environmental Science, Deakin University, Australia. And he's also a PhD candidate at Center for Cellular and Molecular Biology, Deakin University, Australia. He has completed his bachelor's and master's in biochemistry from AMU, India. So welcome, Mr. Hasan, to the program. And so for the ones who are joining us for the first time, I would like to give a brief introduction to what NcodeX is. So NcodeX is a research institution where it was initiated with the aim of bridging the gap between STEM academia and industry. So here at this institution, we have major two arms. One is the research and development arm, the other is the training programs. So for training programs, we have different kinds of programs such as STEM coaching, STEM mentoring, virtual internships, online courses, and many more. So if you are interested in what we do, please have a visit to www.encodexpert.com. So Rubini will paste the link to our website in the chat box. And without taking much of the time, I will move on to the session. So this session, the PhD talk, will consist of two phases. During the first phase, I will let the speaker to do his presentation, and then followed by that, we will have a Q&A session where we would put forward our questions. And if you have any questions related to what he talks and his research field, please feel free to drop them in the chat box. And we are live on Facebook as well. So those who are watching us on Facebook, welcome to the program and be connected. So, Mr. Hasan, over to you.
Thank you. Thank you, Lakshmi. Thank you, Ruvini, for having me. And thank you, NcodeX, for inviting me to this beautiful talk. My name is Qambar Hasan, as Lakshmi have mentioned. I'm currently based in Melbourne, Burwood, Melbourne campus of Deakin University, Australia. Good evening to everyone from Australia, and good afternoon to anyone joining from India, like subcontinent India, Sri Lanka, Pakistan, wherever you guys are joining. Hope everyone is well at their end. I will be talking about, let me just quickly share my screen. I will be basically, can you guys see the screen? Yeah, cool. Yeah, I will be basically talking about things which I have done at my current position, which I was doing my PhD from Center for Cellular and Molecular Biology. I will talk about what I have done in my PhD, what we proposed in my PhD time frame, and basically it will move around. So I should tell that my talk will be more about epigenetics. So I'm an epigenetic researcher. This is for the students, those who are new in this biological research. Those who are already experienced, they know what epigenetics is. Epigenetics is when we actually looking at the factors or any regulatory pathway which controls the expression of gene without being part of our DNA, like having a control over your DNA or gene expression, but it is not part of your gene. So basically any environmental factor, anything which is not inside your genetic material or any of the organism genetic material, but controlling. So there could be 100 types of different things. And I will be focusing mainly about histones and giving you brief introduction and going into the detail of my PhD project. Before, I think Lakshmi mentioned out about myself. So I have done my bachelor's, honors, and master's from India. I'm basically from North India. And I was very lucky to be in one of the pioneer institutes of India, especially the Life Science Research Institute. It's called Aligarh Muslim University. It's like one of the oldest institution, and it got a very, very old department of biochemistry. Like many pioneers have started their career from there. And I was very lucky, and I have very fine professors who taught me those things like basics of genetics, biochem, physiology. And after completing my bachelor's and master's, I was working in India at different institutes. And then I moved to South Korea, and I was doing graduate research assistantship there for 2.5 years. Meanwhile, I was focusing on some novel transcriptional regulatory pathway in muscle stem cells. And then finally, I moved to Australia in 2015, and I got international postgraduate research scholarship of Deakin University. And I started my PhD at the end of 2015, kind of early 2016 in at Center for Cellular and Molecular Biology. Okay, so my talk will be about novel regulatory pathway of Set1 histone methyltransferase in yeast. Our model organism is yeast, but people have been working on Set1 complex. I'm going to talk about Set1 complex in a moment. People work in different set complexes and in different organisms. Why we do yeast? Because yeast is a simple eukaryote, and there are many reasons why transcriptional biologists or epigenetic researchers like us pick a specific protein. I'll talk about that one as well. Basically, coming back to the basic of my talk, again, my PhD project is a very fundamental and a basic research project. It has many applications, but it's one of the fundamental projects where you actually try to build the knowledge. So it's a building a gap in the textbook. So we are trying to find out what actually have not been discovered or what actually have not been found out in terms of different regulatory pathway in protein translational mechanism. I'll talk about that one as well. So I'm going to talk about a histone methyltransferase. Before thinking about histone methyltransferase, let's talk about histones. What are histone protein? Probably everyone from life science background understood what are histones. Histones are basic protein, alkaline in nature, basically positively charged and highly conserved. They basically, the sole function of the histone is to compact DNA, the negatively charged DNA. That's why they are basic or positively charged. They compact the DNA and basically form an octamer. And so compact the DNA and forming a nucleosomal assembly. Basically, all of the chromatin inside the nucleus, half of the chromatin proteins are histone basic protein. In simple eukaryote to higher eukaryotes, we have 5 different types of histones: H1, H2A, H2B, H3, and H4. Out of 5, 4 histones, H2A, H2B, H2, H3, and H4, they basically form an octamer, which compacts the DNA. If you're thinking about, we have a very, very large DNA molecule. That's what I always tell to my student, that even if gonna take out all the DNA, we're gonna circle the whole Melbourne city with our DNA. You know, it's a very, very big molecule. It has to be compact. And when it compacts, then the discussion starts off, because DNA is not now in a compact form. So the transcriptional factor and the protein which has to interact with DNA, they have to find a way to interact. And that is how the epigenetic story comes in. So you got a DNA, any cell got a DNA. How it is going to express, that's a question when it's highly compact. So then the whole story of readers and different types of epigenetic factors comes in the mind. So how DNA interacts with its surrounding or environment basically controls the level of genes in different organisms, from simple eukaryote like yeast to higher mammal like us, like humans. This is just a picture from textbook Lubert's trial, but I'm also trying to put, and if anyone is interested of these information where I'm getting. Basically, these chromatin, when it's compacted, nucleosomal assembly, DNA is compact around the basic histone protein. It's basically formed those small chromatin fiber, which basically forms chromosome when we actually looking at the individual phases of cell cycle, for example, mitosis. So the thing which you observe under the microscope during the mitosis of cell division are chromosome. Chromosome basically nothing but actually a compact or those small fibers of chromatin. Chromatin comprises of those nucleosomal assembly of histones and DNA. As I said, the function of chromatin is not just to package the DNA. It has to hold the DNA inside the cell, but how that DNA is going to interact with different types of transcriptional factor or other factors, that will be basically whole idea will be and comprises the transcriptional regulation of DNA through the chromatin assembly. There are different types of modification, different types of changes that could happen at chromatin surface. And basically, in this picture, I'm just giving you an overview of the 2 meter. It's just an overview, like 2 meter of DNA within the 5 to 10 micrometer diameter of inside the nucleosomal assembly inside the nucleus. So think about 2 meters of DNA you have to pack within 5 to 10 micrometers. So this is how the chromatin or histone compact the DNA inside any cell. One more thing I want to clarify, that I'm sitting in an office. It's not going to be blackout, to be honest. It's just a technological reason. Again, technology has. So I have to move and just move towards the sensor. So it's because there's no one in the office at this time and it's weekend. So if I go like for 5 seconds, please pardon me in advance. It's just technology, you can't teach. So yeah, so the sensor will black out the light if no one is entering from the entrance. Sorry for that. So chromatin, and probably you have heard about these two types of term, heterochromatin or euchromatin. So heterochromatin is basically when the whole transcriptional factor are not able to bind on the DNA inside that nucleosomal assembly around the histones. That's called a transcriptionally silent form of DNA or chromatin. And when it's active form, which means the transcriptional factor can access the DNA inside the cell, it's called euchromatin. So probably anyone have heard about it, but this is how we classify them or we categorize them. I'm going to talk about these ones in detail. Okay, let's talk about histone modification. So what actually happens? So there are different types of modification that can happen around the histones, which can influence or which can impact the genetic or gene expression of the whole organism or inside the cell. And there are hundreds of types of modification. I have just listed few here. And the interesting thing to remember is that its histone proteins are highly conserved. Conserved means if you're going to study about the histones from some simple eukaryote to higher eukaryote, highly conserved. Sequences, the whole structure, or like whatever you're looking at molecular level, they're very conserved, but they are very, very like most variable in terms of post-translational modification. These histones are actually proteins, so they have so many types of post-translational modification, which happens after the translation, and they have so many 100 types of post-translational modification, and each post-translational modification impacts the activity of chromatin or control the genetic factor or gene expression inside any living cells. It's very, very interesting, but again, it's a very fundamental thing which I'm going to target or I'm going to talk about, like how this particular complex is assembled or regulated inside the yeast. So coming back to one specific, so I'm going to focus on, as I said, there are different types of modification that could happen. Let's say methylation, methyl group can be added, phosphorylation, a phosphate group can be added, and this picture actually tells you that which particular amino acid, these are the simple one alphabet for individual amino acids on these stones. K stands for lysine, S for sulfur related. So these different types of modification can basically control so individual amino acid can be methylated, phosphorylated, acetylated, ubiquitolated, ABDE ribosylation. There are so many different types of modification. I'm going to focus on methylation, specifically a lysine methylation, which was mentioned as K in the previous slide. Histone can be methylated, and specifically the lysine residue, because of their individual charges on the lysine, there can be methylated group can be added in single, double, or triple. Like it can be monomethylation, dimethylation, or trimethylation can happen on different types of lysine residues, different histones. Different types of residues, as I said, not only I'm going to focus on H3, histone 3, but other histones can also be methylated by different types of these enzymes, which are called as histone lysine or histone methyl transferases. So histone methyl transferases are those enzymes which can transfer this methyl group basically to those histones and basically methylate them. The function of methylation, or why they are doing the methylation, that is what we are trying to find out. And I'm going to talk about that in detail. That different types of histone can be methylated by these histone methyl transferases, and you will heard about these different terms. So HKMTs are more very common term for histone lysine methyl transferases in simple eukaryotes like yeast. But if you study them, you probably heard about JHT, and they're similar terms. So again, as I said, these individual histones can be mono, di, or trimethylated. And important thing is that so far, up to 2022, what we know that depend on the residues and number of methyl groups added on these individual histones, it can be a mark of active or repressive transcription. So basically, there are different types of, so we cannot generalize that only monomethylation can be a mark of active transcription or trimethylation is a mark of repressive. Just giving an example. So there are different types of methylation, and each methylation has its own impact on the transcriptional activity of the gene. Coming back to Set1 histone methyltransferase in yeast. So histone methyltransferase are overall an enzyme which basically transfers methyl group. In yeast, this particular complex, so in yeast, on H3K4, on histone 3 lysine 4 residue, only one type of enzyme or one type of protein is found which actually does the job. In higher mammal, when we talk about, I'm going to talk about mammals like humans, and in Drosophila, let's say, we have different types of protein. In yeast, you have only one type of protein, which is a complex of protein called Set1C, Set1 complex. It's called Set1 methyltransferase. So you have only one protein in yeast. That is why it's very, very important and very, very functionally easy model to study whole complex in yeast, because you have only one type of protein. You have different types of subunits of that. I'll talk about that one. So in higher mammal, as I said, there are different types of Set1 complexes. And each of these proteins, the interesting thing, why this SET comes in discussion. SET, actually, this is the full form of that. So basically, it's a trithorax domain protein, basically, which is responsible. So each of these proteins, whether it's Set1C or in higher mammal, 6 different types of Drosophila, 4 types of histone methyltransferase, they each have SET domain. SET domain is a specific type of, maybe someone from proteomics background tell you in more detail. Those are evolutionary conserved domains of protein which actually have a catalytic activity. So I'll talk about that in the upcoming slide. So in yeast, we have only one type of histone methyltransferase, H3K4 methyltransferase, which is called a Set1 complex. Interesting thing about this complex, this is probably from my preliminary confirmation document. Set1 complex in yeast, it's a complex of protein and basically have 8 subunits. So it's a one functional protein, one functional complex, but it has got 8 different proteins. I have put them here, individual subunits like as SWD1. We call them SWAD1, and there are different names if you move to higher mammals like humans. Different names for the same protein: SWAD1, SWAD2, SWAD3, SPP1, BRE2, SDC1, SHG, and catalytic Set1 protein. So this is an overview. So this is a complex where individual subunits play an important or individual role to basically in the action or in the activity of the Set1 complex, or also controlling the level. For example, if this complex, let me just tell you one thing, that I'm going to also talk about assembly of this complex, and basically where then actually I will be answering one of the parts of how this particular research is important in terms of sustainability, or in terms of what are the novel aspects of this research. So this particular complex is one of the fundamental, or one of the first complex have been studied or discovered by my supervisor, which have a co-translational assembly. I will talk about that co-translation a little bit later. So why I'm telling you about the assembly is that, for example, in this particular protein, which have 8 subunits, each individual subunit have their own role. For example, if you are missing SWD2 subunit of this protein, the cell or the yeast will not grow at all. So if you are missing SWAD2, for example, I've done so many mutant studies. If I mutate SWAD2, the whole yeast complex will be deassembled. It's not deassembly, the whole cell will not survive. It's an important for the survival of the cells as well. Specifically, for example, SPP1, which I studied in detail during my PhD, are responsible for H3K4 trimethylation. The subunits like SWAD1 and SWAD3 are important for the whole complex to be stably active. So if these subunits are missing, then probably the whole the cell will survive, but the complex will not stably form. BRIE2 and SDC are basically responsible for di-trimethylation. I'll talk about in a little bit in detail. So the idea here is we are going to study this particular complex in yeast in detail, because this is the only enzyme, or this is yeast have only one enzyme to trimethylate the lysine 4 and histone 3. Excuse me. Technology. 15 minutes timer. Sorry. Let's talk about, okay. Let's talk about what actually this H3K4 methylation and Set1 actually has, whatever we have known so far. These are, as I said, it's an evolutionary conserved methyltransferase. And basically, it's important to understand the mechanism of H3K4 methylation on chromatin, like an active form or repressive form of protein. And it has been functionally so far linked to so many different types of process. I will give an overview about them a little bit later. There are different, so this paper, if you want to read about multiple phases, what this Set1 complex can does in different types of processes like transcriptional, chromosomal segregation, transcriptional termination, DNA replication, meiotic recombination. This study was done by my boss back in 2013 in Science. It's I will share that publication a little bit later. So how SPP1 unit of this complex is actually important during the meiotic recombination event in yeast. There are different ways. So coming back to the topic of my talk is regulation of the Set1 complex in yeast. There are different ways through Set1 through which the Set1 complex can be regulated inside the cell. But the problem is we don't know. We don't know how this particular complex in yeast is actually assembled. The reason, let me just skip to one of the thing. Okay, I'm gonna talk about, because probably most of you will be wondering why this particular complex is actually important. I'll go back to that one in a second. The thing is, as I said, the Set1 is the only complex which is responsible for all H3K4 methylation even in yeast. But we have, like for example, in human, we have, I explained that we have 6 different types of methyltransferases, which does the same job. And why this particular complex is responsible? Because most of the children-based leukemia, MLL, AML type of leukemia, this is the protein which is basically a proto-oncogene, which is basically responsible for all of these AML and MLL type of leukemia. And I don't need to tell anyone, like from biological research background, that how important it is to study about those leukemia to find out the drug or assembly. The problem lies in we don't know much about how these proteins are actually assembled. That is why this research is so fundamentally important, that to find out the cure for a leukemia, first we have to understand how that protein actually assembled and regulated inside the cell. That is why this research is very, very like so many of the research group are actually sticking to the Set1 complex in yeast and try to find out the mechanism and different novel regulatory pathways, so that we can have actually target those different regulatory pathway in yeast, and then that can apply to the human as well. Okay, yeah, coming back to the regulation. So there are different factors through which this Set1 complex inside the yeast can be regulated. I'm just giving you an example of the known research. Like for example, H3K4 methylation can be basically regulated through the ubiquitination of histone HB at lysine 123 acid. This is the paper from one of our collaborators from South Korea, Kim et al. They publish in Mol Cells. So basically, excuse me, to control the level of histone 3 lysine 4 methylation, basically ubiquitination of histone HB playing an important role. The same thing they have published that the NSAID domain, the active catalytic domain, basically helps in H3K4 methylation even in yeast. And also we from our group we have found out that SPP1 subunit of this particular protein, which have got this PhD domain. This is again, this PhD has nothing to do with my PhD. PhD is a domain of protein, as in the proteomics background, people know that different types of domains. So PhD is one of the domain of proteins. So this particular protein is actually have a PhD domain, and this basically control the meiotic recombination, how the double-stranded breaks, how that recombination, to form a recombination, you have to form double-stranded breaks on each individual chromosome during meiosis. This particular protein plays an important role during DSP formation, and that is what our group has published in Science in 2013, that in the absence of SPP1 subunit, double-stranded breaks during the meiosis will not happen, and then you have other consequences of meiotic recombination error in the absence of this particular subunit. What we actually try to find out that is there any novel post-translational modification which can control the level of this particular protein inside the cell. So basically you can ask me, okay, you are talking about a post-translational modification, and you are telling me that another post-translational modification controlling this post-translational modification. Yes, I am saying that because there is a reason for that, because the preliminary data suggests that some of the post-translational modification, for example, I'm going to talk about SUMO, summoylation. This is one of the very known post-translational modification. We got to know from our large yeast to hybrid screen that summoylation can basically control the level of Set1 protein inside the cell. So other post-translational modification can control the level of this particular protein inside the cell. Summoylation. Sumo stands for small ubiquitin related modification, which are basically actually like a ubiquitin, and basically added like a ubiquitin, how the ubiquitin translational or ubiquitin targeted proteasomal machinery works. Similarly, they target individual proteins and basically degrade them through the proteasomal degradation pathway. Same way like the way ubiquitin basically targets the individual protein. In our case, we found out that from the global screen, I'm going to show that data in a moment, that two of the individual subunits of this particular protein have actually have a sumo interactive domain. So basically those two individual subunits can be summoylated inside this particular complex. And when we were actually screened the whole complex from yeast to higher mammal, we found out that not only three different types of protein basically have interactive domain present. Like basically they are interacting with one of the protein, which is a sumo protein, a NIS1 protein. And this NIS1 protein basically, NIS1 protein is basically a part of sumo-based regulation of the different protein or proteasomal pathway. They have a specific domain where individual on the C terminus basically, which basically targeted by sumo targeted ligases, which basically degrade these proteins through, and this is how the protein level is regulated inside the cell. So I'm not going to discuss in this particular slide in detail because it's going to make things complicated. Again, the idea here is that how this summoylation, a post-translational modification, is controlling the level of Set1 histone methyl transferase level inside the cell. This is what my PhD, like one of part of my PhD was. Is there any functional link? Is there any correlation, or is there any specific protein, or is there any specific interactive partner which can interact or which can basically target this particular the specific subunits of this Set1 complex and control the level of Set1 complex? As I said, I'm going to quickly talk about the assembly of the Set1 complex. The reason is, well, I want to tell, I just don't want that audience, probably if they are interested to know more about Set1 complex, that why the Set1 complex is also important in terms of assembly pathway of the protein. Okay. Our group in 2019, my supervisor and his group, before like I joined this PhD, they have found out that this particular protein, Set1 protein, is assembled co-translationally. When I say co-translationally, because that was part of one aim of my thesis was to find out what is the functional significance of this co-translational assembly pathway. Before I talk about that, I'm just going to just say that when I was studying, or probably when you guys were studying, whoever is listening, probably you know that most of the proteins, like the active protein, whatever protein any enzyme you talk about, anything in any living system, most of the proteins are actually a multi-protein complexes. Protein rarely work in isolation. You hardly found a single protein doing everything around it. So proteins basically exist in a complex form. Like each complex have individual subunits, and then basically they form a complex, and then that whole complex have individual subunits for individual role. I'm talking about from simple eukaryotes, from bacteria to higher mammals like humans. The interesting thing is that when I was studying, or when probably even so far, the textbook, any textbooks of biochem, I've got so many books on the bench. Each textbook will tell you that proteins, each protein or each complexes are assembled post-translationally. The meaning of post-translational assembly is that each individual subunits are synthesized on ribosome, translated, and then they are going to find out a partner around them, and then they assemble. There's a gap. That's something. There is a big gap. No one has ever. So that is how, because there was a big gap between a knowledge gap, that how that is even going to be possible. If we talk about, let's say, talk about yeast, millions of protein are basically synthesized on ribosome in a fraction of second. Millions of protein. Let's talk about simple eukaryotes. How individual subunits are going to find out their partners around them if there is no kinetic or there is no mechanism. This is how the group, like our lab group, my supervisor and his associates, found out that in yeast, this particular complex, it's not translated post-translationally. It is a co-translational assembly, and I specifically want the young audience, specifically who are doing masters or bachelors and interested in research, think about or just try to find out what this actually is, because this is one of the universe where you can think about co-translational assembly of protein. So they challenge the whole notion that proteins are not actually like a translate or assemble post-translation. Each individual protein cannot find its partner in a surrounding. It's like a jigsaw puzzle. How a protein is going to find its partner if they don't know what is happening? So my supervisor, they publish in EMBO-J that this particular complex inside the yeast is actually assembled co-translationally. They were not the first person who found out co-translational. There are many bacterial protein were discovered before, and after that, hundreds of protein have been found out, or complexes have been found out, that they are actually translated co-translationally. The meaning of co-translation is that when each subunit is on the ribosome and they are about to finish their translation, the other subunit basically start interacting with it. So it's not like the whole translation will happen and then they will assemble. So on the ribosome, when they are on the ribosome or the polysomal fraction, that's how I've done those polysomal analysis, that each individual subunit, when it's about to finish or going through its translation process on the ribosome inside the centriole, they start interacting with their individual partner, and then they start assembly along with the translation. The reason, and my when I started my PhD, one of my aim or one of my project was to find out why that is happening. What is the force which is driving the cell to go for co-translational pathway rather than post-translational pathway? And that was one of the most challenging project I have ever found, like I've ever worked with. So basically, we had a sort of mechanism or hypothesis that it is the basically a sort of this is an overview what we have found out, that it is the concept of kinetic completion. The reason is when individual subunit or interactor, the one which has to interact with individual subunit, is missing, then basically instead of proceeding to form the pathway or form a complex, the proteins are tagged by ubiquitin and then basically degraded through the proteasome. If an interactive partner is present around the ribosome, then the whole post-translational assembly pathway will work, and then the translation will continue, and basically productive complex formation will happen. So this is what we propose, that it is actually the feasibility or the availability of the partner which derives the cell to go for co-translation. Because otherwise, if you think about, it is the wastage of energy, it is the wastage of time for cell itself. Forming an individual subunit, and then subunit is not able to find out its partner through which it has to interact and form a complex. Anyways, I'm not going to take a time of this because this can take ages to, because this is one of the very like a fine discussion. Again, this is a textbook based research topic where you can find out the difference between co-translational and post-translational assembly pathway. I think I have talked about that. What is the significance? Just briefing it that this is in terms of assembly, this is one of the fundamental or fundamentally studied protein, which tells you about how proteins are actually assembled in any living organism, starting from eukaryotes, simple eukaryotes like yeast. And as I said, MLL and AML type of leukemias in children, each of these protein are actually Set1 based protein in higher mammals. And we don't know much about that, how those proteins are actually assembled, how those proteins are actually regulated inside the cell. That is why studying Set1 complex have so many of the applications in applied research. So when we proposed, or what are those different questions, basically when I started my PhD back in 2016 and like kind of end of 2015. So basically I was trying to find out what are the different types of novel transacting factors which can basically target Set1 complex and individual subunits and basically regulate the level. Okay, so basically trying to link the H3K4 methylation, because that is what this particular complex does, H3K4 methylation, and basically trying to find out is there a functional link between this H3K4 methylation, which is performed by this protein, to the other cellular process, so that we can find out novel regulatory interactors. And once we have novel regulatory regulator, we can target those regulator and think about how they are interacting with Set1 complex, and basically what we can target in terms of application. For example, if we're thinking about ML or AML type of leukemia and their drugs. Okay. The second part I just discussed, that of how this particular complex is assembled is co-translational, but why cell is preferring a co-translational pathway than a post-translational pathway. This is the whole hallmark of my supervisor's last 10-15 years of it. He has been in this field for the last many years, but like from last 10-15 years, he's focused on why basically, and you probably will not surprise that textbook will be changed. And I don't know when, but textbooks, specifically those biochemistry or those molecular biology textbooks, have this thing which they will explain that cells are not actually preferring post-translational assembly, they are preferring co-translational assembly. Once we have an actual answer why actually cell is going for co-translational, because to prove or disprove because there's no proof of post-translational, but that has been proposed by the knowledge. But we have to, if you have to disprove that, you have, or universally prove that, you need more complexes or more proteins which are co-translationally assembled. Anyways, in terms of research plan, I had different types of research plan. Basically, aim 1 was the novel regulatory pathway, aim 2 was find out the assembly of Set1 complexes. So in first part of my regulatory, or aim 1 on the left hand side on the slide, I was trying to build, we were trying to build actually extended Set1 interaction network, basically based on one of the finer technique, which called yeast-2 hybrid. So basically when you try to find out the interaction between two hybrid, I can talk about the yeast-2 hybrid, but it's just one of the tools when the protein interact. People who are working in proteomics, they can tell you when you have to find out which protein is actually interacting or not regulating. You do different types of assay. Yeast-2 hybrid is one of the standard biochemical assay where you actually try to find out novel regulatory interactor of any protein. And once we have an extended protein interaction network of the Set1 complex, we can think about post-translational modification. For example, in my case, I was very interested with simulation because we have many 2 or 3 protein of Set1 complex, or 2-3 subunits of Set1 complex are actually interacting with sumoylated protein like NEAS1, which have sumo interactive motan. So I was actually trying to find out whether actually there is any summoylation or this Set1 complex or any subunits actually have summolation event happening, so that I can know that if that summolation is happening, then how that is controlling the level of protein. The third point, or the third part, which I have mentioned here, impact of copper, I will talk about that later. Um, this is this thing was never a part of my PhD. This is what happens. Those who are doing PhDs, those are in research. This is excuse me. Qualities again, technology. We need AI, to be honest. So this was one of the observations when I was doing this when I was doing this particular regulatory project. I found out a very interesting phenotype. I'll talk about that a little bit later. And this become one of my major project of PhD, basically. And to be honest, that was the most interesting thing I have ever, or my supervisor has ever noticed. I'll talk about that later. And this was never a plan, to be honest. And I have written down there because I wanted to tell about that one, but I will talk about that, how this copper story comes, because you will be thinking, this guy has not talked about anything about copper. Why he's talking about copper and affecting the level like H3K4 methylation. I'll talk about that later. Okay. In terms of second aim, again, as I said, co-translational assembly pathway. So basically trying to find out what are the different requirements of Set1 subunit for the in terms of co-translational assembly, and how I'm saying that the proteasomal degradation of Set1 is happening. For example, interactive partner is missing during the translation or on the ribosome. So basically, it looks easy to be honest, but when you actually doing polysomal analysis or working with those around the ribosome, so basically trying to find out which partner is missing, and then in the absence of which partner, the whole basically the complex is not assembled properly, and basically degraded through the co-translational degradation of Set1 happening. So in terms of regulatory pathway, as I said, building an extended protein interaction network. This was our so we basically build an interaction network because of many reasons related to, because we are still trying this research is undergoing. I cannot show many of those yeast to hybrid screen. I'm just showing just an overview. So this is an overview. So this particular complex is a complex of 8 protein, and we try to build an interactive network based on the yeast to hybrid data, yeast to hybrid screen. And for example, this particular one, I'm more interested about this SPP1 protein of this Set1 complex. And we have found out that it basically interacts with many proteins, and basically this different, the color coding here represent how basically strong that interaction is basically. And those who are far, and those who know the yeast to hybrid, those who are far and without the color code, that means they are weakly interacting with those. Anyways, the idea here was that find out which particular subunit of this complex interact with different types of known proteins, and then basically making an overview what we can target. So we found out from our different screen, I'm just showing you one of the screen here, that one of the protein, which is a sumo interactive protein, NEAS1, is basically interacting with 3 different subunits. And this is how then basically I started to basically targeting these 3 different subunits of this particular complex, that these 3 subunits of this complex are basically interacting with this NEAS1 protein. This is an overview of NEAS1 with respect to summoylation. So this is a sumo-based summoylated protein, basically, which have a sumo interactive domain towards its C terminal, basically, and this binds to the SMT3 or sumo interactive domain. And this SID binds to the SMT3 and then regulate the level of NEAS1 and other protein. So idea here was to try functionally link this summoylation to Set1 level inside the cell. Thus, this summoylation has something to do with the Set1C level inside the cell. And if does, then what is the role of this particular protein? Because this particular protein, NEAS1, is interacting with 3 different subunits of this protein. So have done many experiments. I just try to make an overview what we have done. So trying to basically find out those interactive partner, like these three individual partner, SDC1, SPP1, and SHG1. These are three subunits of this particular complex, Set1 complex. Okay. What I did, I just try to overexpress this particular protein NEAS1 in those background of those yeast cells, and basically I basically overexpress the SUMO overexpression in these cells in these background, and basically trying to find out if I overexpress the NEAS1 in these cells, how what are the level of this particular Set1 complex, or if specifically these individual subunits are the level of these individual subunits are controlled inside the cell, or is there any direct impact of histone methylation by the overexpression of these NEAS1, or let's say SUMOylation. So the experimental part has been done. We are in the process of trying to find out that this NEAS1 and some we found out other protein as well, that which is responsible for SUMOylation is interacting with these individual subunits and basically controlling the level of the level of histone methylation inside the cell. Interesting thing, we have found out that when we are actually doing with that, because we have got collaborator, this work cannot be done by one group. We have 4 or 5 collaborators, one from South Korea, I think we have mentioned we have collaborators, and 2 collaborators in Canada, 1 in France from CERN. So they have found out that even though we express SUMO, SPP1 protein has not been found out that there is any SUMOylation happening around SPP1. Then I stick to the other 2 proteins, SDC and SHG, and I have done the SUMOylation study of overexpression of SUMOylation on these 2 subunits and try to find out. Okay. I'm also going to give you an overview about the third aim of my regulation pathway study of Set1 complex. As I said, there are some limitations, specifically with these open talks. When the data will be published, you guys will have more what we have like actually discovered, because some of the data cannot be shared on open spaces until it is being published. And unfortunately, we are still doing some experiment, and we are still doing some analysis in terms of SUMOylation. Okay, when I was doing this overexpression studies, and to be honest, it took me almost like 2.5 years just to establish those overexpressions and just to find out what I have just explained, that these interactive partners and these are interacting. This take ages to just establish those techniques and those understand that there are 2 or 3 interactive partners of this Set1 complex which are interacting with SUMO, let's say SUMOylation, probably. So basically, there is one way of doing an overexpression. Those people who does overexpression of different proteins, they know there are different tools of doing it. One of the tool is that metal-based transcriptional factor, which can be activated just by adding simple like copper sulfate or like some other sorts of copper. So basically, when you're trying to overexpress any, like when you are trying to overexpress any protein, basically, you basically for example, well, let's talk about yeast. Basically, you transform that cells with certain plasmid, which have your, basically you clone your particular protein, you clone your gene in there, which is basically can be overexpressed by different promoters. So idea was that we have this Set1 complex, this individual, the SUMO protein under the control of copper-based promoter. So idea was if I add copper in the cells, the copper will control the level of those promoters, and then when those promoters will be actually active, then the downstream on the downstream of that gene, I have my own SUMO-based protein, overexpressed protein. Those proteins will be overexpressed inside the cell. The interesting thing, and like I wanted to tell you how that was actually. So when I was doing those, so I'm talking about 1.5 or 2 years of my PhD, I was doing those overexpression, but never noticed. But later phase of my PhD, I started noticing one thing, that whenever I was overexpressing my cells, was trying to do any overexpression study using this copper-based transcriptional factor, copper-controlled transcriptional factor, one of the protein of this particular complex, Set1 complex, is always migrating faster. You can see that this is a Western. So this protein, in the absence of copper, you will find the band just here, like let's say. But whenever you add a copper in the media to overexpress, because the overexpression will not happen because this is under the control of metal-based promoter, this protein always migrates faster on the, this is a SDS gel. And even in the Western, you'd see a signal like that. This is a Western signal. And not only that, the interesting thing was that the methylations, because that is the function of this particular complex, Set1 complex, the trimethylation is always increased by many folds in the presence of copper. So in the initial idea, whenever we were doing experiment, was that okay, this has to do with the simulation, because we are trying to overexpress simulation. But then I just instead of using those sumo-based promoter or plasmid, I just simply treated myself with just like a control plasmid in the absence of any of those proteins, and I found the same result. That trimethylation is always high in the presence, and then I did so many experiments, like this is just a titration experiment, different concentration of copper from 0 to 500 micromolar, and the protein, this particular protein, for example, I'm talking about SPP1 protein. This is a subunit of Set1 complex. Anti-TAP here means because each of these proteins have a tag, TAP, tandem affinity purification. We purify through these tags, so we don't have antibody against SPP1. We don't have antibody. When you don't have antibody, though, in the molecular biology, there are so many limitations. One of the limitations with the people like proteomics or transcriptional people like us is you do not have antibody against many proteins. So you have to basically tag that protein with certain tag, and then you have an antibody against those tags, and then you basically discover, you can basically find, you can discover or you can find out that signal on your Western or on the protein gel using those tags. So please do not get confused with what is this TAP. TAP is just a tag. So I was telling that, so initial one, like when we were doing simulation based overexpression, that this has to do with the simulation. But later on, I found out that this has something to do with the metal. So then I tried to find out literature, and there was nothing in the literature which suggests. So this is how this part, because what is the impact or what is the effect of copper on H3K4 methylation? Because this methylation event, for example, we are, let's say, yeah, no, I did not put another slide, so I just wanted to show you something else. But again, this trimethylation is a hallmark of repressive transcription. Again, in particularly with respect to H3K4 methylation event in yeast. So if this H3K4 event, guys, if I'm going over time, please let me know. I will have to make sure that I'm on time. I know I'm like taking too much time. I will be quick. I think I should be done in next few minutes. This H3K4 methylation event or trimethylation is a hallmark of repressive transcription. So then we got a very interesting or intriguing, like me and my supervisor were very like that day was the most excited day. And we were like, oh my goodness, this is the thing. So no one has ever thought about metal toxicity in terms of transcription. So the idea here is you're putting too much copper in the cell, and this cell or the chromatin is basically actively transcribing or actively trimethylating histone to shut down the transcription. It looks simple, but to be honest, yeah. So idea here is metal toxicity is sensed by those these histone methyl transferases, and basically signal the cell to shut down the transcription. Otherwise, the DNA will have damage, and no one has ever thought about. So this is how my overexpression study or simulation study became one of my PhD projects, whether like impact of copper on H3K4 methylation. And we are still trying to figure out many of the things with that. And in 2021, I think I discussed that with Ruvini in the past when we were teaching together, that one of our collaborators has published in Science that there are potentially copper binding sites on histone 3 and histone 4 tetramer, and that has potential copper binding site, and that is how they are controlling the level of those potential copper binding site are controlling the level of other post-transcriptional modification. So our idea is also being supported by them, but we have got too much detail of that, and we actually trying to find out the actual mechanism, how this metal toxicity is sensed, and is there any biochemical characterization of this particular protein with respect to H3K4 methylation event. Anyways, this is how this particular project gave me another project, which became the main project of my PhD. Okay, I'm going to just quickly an overview. That will probably be my last slide, because I just don't want to take too much of time. I was talking about the assembly pathway, co-translational assembly pathway. Unfortunately, because of the time limitation, and those who are doing PhDs in places like Australia or Europe, they know that we have a specific time frame to complete the PhD. I could not go ahead with my assembly project. There are so many things which I have to do, my supervisor has to do, but because of the time limitation, I have to narrow it down. So the idea was, the task which was given to me was to find out the functional significance of co-translational assembly, like why cell is going for co-translational assembly pathway inside the yeast cell or any cell. So the idea was that in the absence of interactive partner, these cells will not go for assembly. They will basically degrade it through the proteasome. And this was a basic preliminary experiment which I have done, or which we have done, that Set1 is basically unstable in the absence of key interactive partner. And you can see that, if you remember, I was saying that the SWAD1 and SWAD3, those are the protein which are responsible for the Set1 assembly or Set1 stability. In the absence of these proteins, SWAD3 and SWAD1, you can see this is a Western blot. When these two proteins are basically absence on the inside the cell, or inside basically around the Set1 complex, the whole complex, the Set1 complex, is not basically going to be stable, and it will be degraded through the proteasomal. Why it's going to degrade with proteasome? You can ask me why you are specifically mentioning about, because I was using Mg132, which is one of the inhibitor of proteasome. So when you treat cells with Mg132, which is one of the compound which basically inhibit the proteasome. When it inhibits the proteasome, the meaning of that is whatever is happening through proteasomal degradation pathway, that will not happen, because you have inhibited the whole proteasome by adding this Mg132 compound. So the idea was, when I add Mg132, these two interactive partner, which is basically responsible or required for the Set1 complex to be stable, needs to be when there is when in the presence of Mg132, the Set1 protein needs to be a little bit stable. But the data was not very promising, that although the Set1 complex, the idea is correct that Set1 complex is not stable, but by just degradation of adding the Mg132, the proteasomal degradation pathway or the overall degradation cannot be inhibited. But still, the whole complex is unstable and going through degradation, but we don't know through which pathway. We were targeting proteasomal degradation, that there could be other pathways that can be degrading the whole protein. We are still going through this whole project, and there are so many things still need to be figured out before answering the question why, or answering that thing or adding this thing in the textbook, that co-translational pathway is the actually fundamental pathway of protein assembly pathway, not post-translational. So I'm just going to skip this slide, because this has gonna talk about co-translational assembly pathway. I think I have mentioned in detail. So we were doing polysomal analysis, some assays called a ubiquitin sandwich assay, basically trying to find out whether the cell is going through post-translational or co-translational degradation pathway. So basically, some of these things have been done. Some of them could not be complete because of time limitation, but we will be doing that in future as well. Overall, I know things were complicated, specifically this talk, I know. But overall, this particular project or this particular thing which we are doing in the lab basically give an overview about the Set1 complex and its novel or extended cellular protein interaction network. And these novel interactive partner, for example, I'm saying summoylation, will give an overview about how different or unrecognized functional pathways are linked to this particular methylation event. And interestingly, the one which I found out that our serendipitous observation that copper treatment on H3K4 methylation can discover a direct link between heavy metal stress and the regulation at the chromatin levels. The reason is because whenever you read any metal toxicity or any paper with any cells or any organism, you always talk about toxicity, antioxidant, but no one has ever think about that metals toxicity can be sensed directly by the, excuse me. Metal toxicity can be directly sensed by the chromatin at a very first place to shut down the transcription. So that was our observation phenotype which we found out, and we are still like doing some further experiment to prove that this particular or one particular subunit of set complex is basically controlling the level of copper-based toxicity, or how that particular protein is linked to copper-based toxicity and regulating the transcription level. Okay. Finally, we still trying to figure out about the co-translational protein interaction, that how that pathway is being regulated and why cell is basically preferring. And then if the cell is preferring the co-translational pathway in terms of assembly of protein, then what is the basically role of that in terms of surveilling the whole complex assembly? I would like to extend my thanks to my supervisor, my friend Neil, who was a postdoc, Shashank. He's currently a postdoc at Florida Institute of Neuroscience. He helped me a lot with specifically because I had no clue about the metals and stuff. And all other people of my school, CCMB, the whole school, and Deakin University. And thank you, NcodeX, for inviting me. It was really lovely to talk with you guys and interact with you. And these are some publications, if you are interested, some of the things which we published so far, which, like, for example, this paper from genetics, which I was a co-author of that, we published related to that H3K4 methylation is basically responsible for coordination of cell cycle progression and mitotic spindle assembly. And this is the papers which I was talking about, paper number 2 and 4. If you are actually interested about co-translational, please read through these two papers. They're very interesting paper. That's a new different word of understanding that protein complexes are assembled co-translationally. And paper number 3 is the one which I was talking about. In the science, our lab has published that H3K4 methylation, specifically SPP, one of the subunit, is linked to the meiotic recombination event. Thank you, and I'm open to any questions and feedback, suggestions.
Thank you, Hasan. That was a very insightful talk. I actually liked the way that you started from the very basics and then you gradually went into this technical stuff. So let's get to questions. I think we have a few questions from the audience. Let me read them aloud.
Sure.
Yes. So could there be any genetic or transcriptional influence for subunit assembling post-translational assembly process?
Sorry, okay. Sorry, could you please repeat?
Yeah, I put it in the chat as well so that you can.
Yeah, I'll just stop sharing so that will be easy for me to read. Could there be any genetic or transcriptional? Yes, you are 100% right. Yeah, I think whoever has asked, I really appreciate this question. Yes, there are so many genetic factors. There are so many genetic actually things which people have trying to find. So as I said, my boss was not the first person, otherwise he would have got Nobel Prize at co-translational pathways. But interestingly, he was the first person, or his group was the first person, because he's from EMBL, European Molecular Biology Lab. He moved to Australia in 2011. So he published, or he found out this thing back in his lab in ETH Zurich. This co-translational pathway in eukaryote, because in bacteria you can find, because when we talk about the polysome or ribosome translational process or assembly process, in bacteria, it's because it's a prokaryote, it's different than the eukaryote. They were the first one who found out that yeast, this can happen as well. So, and that is what this whole thing is. How different types of other factors which control the transcription can basically control this whole co-translational assembly pathway. To be honest, we are still trying to figure out. There are so many other factors and there are so many limitations of this field as well. But I'm open for any discussion if you have any particular suggestion with respect to that.
Yep. Thank you, Hasan. And there's another. I just now pasted in the chat. Could there, no, apart from leukemia, which organs do we find these pathways more frequently? So that it's helpful to extend the applications, and can we use in this similarly, can we use any other mammalian cells?
Of course, you can. You should actually, if you are. I think because again, in the start of the discussion, as I said, the kind of stuff or the kind of research which we do is a fundamental, very basic. To be honest, it's a fundamental, basic research. There are so many things in the textbook. These are called knowledge gaps, which we don't know. There are people who are doing fundamental research. Fortunately, we, like my lab group is one of the group which do fundamental research. There are many research, fundamental, secondary, tertiary. Those are very, very important. But there needs to be some things at the primary or fundamental level. We have found out these particular things because, as I said, we are targeting Set1 complex because Set1 complex is only one protein which is basically responsible for all H3K4 events. But in higher mammal, like for example, Drosophila, you have 4 types of protein which does the same. In mammals like humans, we have 6, 7 types of these proteins. You can target and you should basically, if you are a researcher, I would say that. And I'm 100% sure I have read so many papers since 2009, since my boss has found out this thing in yeast. So many people have found out that many protein, many specific, for example, you're trying to talk about different organs. Yes, different types of proteins are basically assembled co-translationally. And the thing is, we are still, because the thing is, you cannot remove those knowledge from the textbook as just by 10 or 15 years of research. We still need to make sure that this has happened ubiquitously, like unanimously everywhere. Okay, so the data needs to become, the research has to be done. But you can apply, you can extend this application to any of those proteins. If you are in a proteomics, if you are doing those assembly, studying different pathways of different proteins, you can apply that. As long as you understand or you have an hypothesis or you understand that why this part, for example, if you're targeting this particular tissue inside the particular organ, why this particular protein will be basically co-translationally assembled. Again, the concept which we proposed was kinetic competition. Kinetic competition means, let's say, let's challenge the notion of post-translational modification. Each protein, let's say talk about the protein which we are studying. It has got 8 subunits. Each subunit, so the textbook says each subunit will be translated on ribosome. So once the translation will be done, then they assemble. Well, again, as I said, you got millions of proteins synthesized every second, okay, like fraction of a second. How they're going to find out? This is how the whole idea of kinetic competition is. If an interactive partner, which is still on the ribosome or on the polysome, because we analyze polysomes, on the ribosome itself, it's not finished its translation, if interactive partner is around, they start interacting and assembly pathway start. If they are missing, then that particular subunit will be proteasomally tagged by the proteasome, ubiquitin will be added, and then the whole complex will be degraded. Because, and I'm just saying this is our hypothesis, or this is our experimental, so far whatever we have found out. The reason for saying that this is a kinetic competition. The cells do not want to spend so much energy, so much time just to translate something which is finally has to be degraded. So there needs to be some plausible explanation for or concept which can explain post-translational, then co-translation or co-translational over post-translation. You can apply to any cells. Thank you.
Yeah, Hassan. So about NcodeX, you know that NcodeX is highly focused on innovation. So, with regard to your research, how could you bring in innovative aspects?
Well, to be honest, our project is totally innovative in a way that
Yeah,
we are fundamental research people, primary research people. It's considered as, to be honest, it's considered as dry field. Many people do not like specifically the place where I'm doing research. Primary research are not very funded. Because I worked in some Asian countries, some of the Asian countries, specifically Japan. Japan fund primary research a lot. Australia, to be unfortunately, do not fund primary research. They're more. I'm not saying that tertiary or secondary research or applied research is not important, but fundamental research is as important as of. And this is how I some people. So whatever we are doing is totally novel and it has so many applications. I'm not saying that this is limited to one. If we have an answer that this particular complex, Set1 complex, is assembled and this has regulated through this pathway, then we can apply this approach to any of the MLL, AML type of protein, which is a basically proto-oncogene responsible for all types of leukemias in children. The reason why we do not have so many drug targets or we have so much knowledge gap is because we don't know at the first place the Set1 complex, which is the fundamental or basic complex in yeast, doing the same job, how that protein is assembled and regulated. So this has got so many different types of novel aspect, I would say. You can take this research to any level, whether you are doing drug targeting, where you are basically looking at novel inhibitors, specifically AML and MLL type of leukemia, you can target any, as long as we have some fundamental data or fundamental knowledge to say that this particular complex this is how this regulator assemble.
yeah, thank you, Hassan. So finally, we would like to ask you about the advice that you would give the young people who are interested in maybe pursuing a scientific career, maybe doing a PhD, maybe doing their higher studies in the field. So, what would be your advice for them?
Like I would highly encourage everyone who is doing bachelor's or master's, if they want to be in research, try to find out your interest and basically stick to again this thing I tell to all my students. I know some of people who are listening will be senior to me, researchers, but if this advice is for the undergraduate specifically, and like early undergraduate or like masters, stick to your textbook. Please read through textbook. I know we are we are back to with online systems and everything is available on internet and specifically presentation, but I still got, if I can show you my desk where I still got 4 or 5 textbook. I still read Leninger. I still read Stryer. Okay. To be honest, the first thing you want to have a knowledge of research is to have a very, very good or grasp of any concept of anything. You cannot just say that I have studied about translation from a slide. You need to study about translation in detail. I'm saying that core translational, post translation. First, we have to understand how different types of transcriptional or like say, how different types of pathways of translation in bacteria versus eukaryote. So please stick to your basics, and I will highly encourage anyone who are interested in research is that try to have some research background, whether it's voluntary, involuntary, like paid, doesn't matter. Try to start working with some industry, some research lab. Try to talk to your professors. If you are interested in research, I understand research is a very long career, to be honest. You have to do masters, you have to do PhD, sometime even after PhD, postdocs, academic career. But if you are inclined towards research, you should go for it. I know sometimes people will be saying, "Oh, I will be not earning as much if I end up in industry." I know, but still, we need researchers. We need specifically biomedical research. There are so many things which have not been discovered. You have a chance. You have time when you are studying, and stick to your basics. Stick to your textbook. Talk to your professors. You can find many people around you if you are in uni, your lecturer, your professors who might take you as an intern or something. So start your research early, like basically try to have some idea about how that, so that will also give you an idea how a research career would look like. Probably you will find out that, okay, I am interested in research, but this for example, this particular research is not into me, I'll probably need to go to the tertiary research or applied research. So there are different types of research, as I said, primary, secondary, tertiary, like fundamental or applied research. So you will, and again, the last thing which I would say is that anything is not the end of the day. Okay, wherever you are, you'll always find other ways. So even if you are interested in something, you will not find probably a PhD position in that field. Grab your opportunity. Probably you will have more opportunity in that field. So for example, when I was doing my bachelor's and master, I will stick to this one and I will be with that, sure, I will be in this field. And I was doing my jobs and stuff in mammalian stem cell biology back in South Korea and those places. I was like, I have to stick to this one. But then I found out this yeast genetics, and I was initially in the early days of my what I'm doing, why I moved to this one. But now I found this is a totally different world. So do not confine yourself. Do not be biased. There are different types of research field which you can go, which you can opt, and anything and like at any place, it's not the end of the world. You can always have more opportunity, more option, and you can switch field. I know people, or even I'm interested to do something differently. Like I want to do my second PhD in some other disciplines. So I know people who have done PhDs in 2-3 different disciplines. I'm not saying everyone should do that, but you will find out that people have different types of inclination, different types of interest. So do not just confine yourself to just stick to one thing. You can have more opportunities and things when things goes on. And yeah, happy to take any other questions. Yeah.
Thank you, Hassan. Yeah, that's it for today that we have. So before wrapping it up, Rubini will paste the links to our feedback form and the other form which you can fill it up, the audience, the people who can fill it up. And if you are interested in taking part in one of our programs as a speaker. So Hassan, thank you very much for being here today with us, and that was an excellent talk. And I thank you for sparing your time and sharing your knowledge with us. And I think that was really inspiring as well to many of
Thank you
us.
for having me. Thank you, Rovini. Thank you, Lakshmi.
Okay, thank you very much. So see you guys soon. Till then, take care. Bye bye.
Yeah.